Next speaker, Dr. Yani Sofra. Yeah. She has a doctorate of neurophysiology from London University and clinical psychology from New York. And 3 books be translated into 9 different languages. She's written around 73 scientific articles in the subjects of neurophysiology. So let's welcome, she's going to present the collagen miracle and why it's not enough for useful rejuvenation. So let's welcome.
Thank you very much, Dr. Peng, for the wonderful introduction, and thank you for having me in Taipei AMWC. I'm honored to be here. We're going to be talking about the collagen miracle. Everybody knows about it, but why is it not enough? So I have no conflicts of interest in presenting this lecture. Now, aging will speed up the disorganization, the body's dysynchrony. So we need a solution. And here comes the anti-aging industry and presented to us the collagen miracle. And what is the collagen miracle? As you can see, the collagen there is, intact. And here, both collagen and elastin, they start getting deteriorated. And same thing, the young and the aging skin have the same problem. Now, there is one method, the energy devices, because the trauma will increase collagen. The problem is that you see here, there is not enough collagen in the body. So if there is not enough collagen in the body to produce it, then what happens? There is also some caution with very young bodies that you could have some side effects with keloids, because that's what collagen is excessive collagen creates scars and keloids. And the second method is what I call the evaporating liquid method. So it's like, aging is like a glass of a liquid that evaporates. And so we need to fill it in again. So we put more stem cells, so we put more hormones, so we put more collagen in this particular case to replenish it. Now, there is a little bit of a side effect here. It's not going to happen all the time, rather rare, that collagen may create what we call hypoxia. It might kind of obstruct the oxygen that circulates inside the skin. And that might be a problem. But let's start from the beginning. What is collagen? Collagen is a protein. So this protein is made of amino acids, about 20 amino acids that you can see there. Actually, I got this slide from the 2024 Nobel Prize research. I'm going to talk to you about this later. And this is what will kind of coming down the pike eventually. The most important thing is what kind of proteins can we create to reverse aging. So here is the problem with collagen, just collagen, basically. Only in the epidermis, there is about 18,127 proteins. And we need all of these proteins and then interaction, I will talk about the interaction a little later, in order to have this anti-aging rejuvenation effect. So you have 8,987 in the outer epidermis and 9,140 in the inner epidermis. Just on the epidermis, we have about 19,000 plus proteins. And this is basically the research. And here is the famous Nobel Prize 2024 in chemistry. And what did they do? This is the scientists that got the Nobel Prize, and they discovered 200 million proteins in the human body. And they said, well, that's not enough. We need more. And they discovered the first protein. This is the first protein that came out of this computer program, because they basically found out how the proteins are structured. So they cracked that code of the protein structure. And this is what's going to become 7 years 5 to 7 years later, you're going to see everything is going to be about protein and proteins and new proteins that have been formulated to reverse aging. And this is some of the other proteins that they basically created. New nanomaterials, about 120 proteins, kind of how they kind of interconnected. And put a pin on that word, interconnected. Why? Because it's a very, very important word. Because it's not just one protein, it's how proteins are connected that makes the difference. Not just collagen, but 18,000 only in the epidermis and 200 million in the human body. And this is all the new proteins. You can see the 2017, they found how they can basically use it to defect fentanyl and other addictions and other nanoparticles that can be basically as part of a vaccine and so on and so forth. And this is more proteins, and how, again, what they studied is how these proteins intertwine to kind of form like cities or kind of bundles of proteins that are for a specific purpose. Now, the other problem is what I just said, to put the pin on. Replacing one protein or even 200 million proteins is not enough. Why? Because all these proteins interact. Proteins are the brains of the cells. They're the alive part of the cells that basically is the communication. Exosomes, you talk about exosome. What is exosome? They carry proteins along with the microRNA and the mRNA and lipids and nucleic acid, etc. So filling up a gap does not mean that you can reinstate communications between the proteins. And that's the important thing. And what you see here, you see how the two cells are communicating. You see how what's happening there is an interaction. This is all proteins, how the cells communicate between them and create. And this is under the microscope. So proteins are the brain of the cells, and the cells make a communication system. And youth is the result of optimum proteostasis, optimum balanced proteins communications, not just proteins. And we need all these interactions and intercommunications between proteins to rejuvenate, to create the anti-aging effect. So, and there has to, you see, out of sync, okay, in sync is going to be a faster communication. These proteins have to be in sync, and I will talk about resonance shortly, how it's very important for the communication in between cellular communications. Now, this is the Nobel Prize in Physiology and Medicine 1999, which found that the proteins are carrying labels like addresses, that they know exactly where to go from the time they're created to where is going to be their function. Like you go to a job and they say you kind of going to be doing shipping, you're going to be doing interactions, marketing or whatever. It's the same thing with the proteins inside the cell. That basically, this is what the 1999 Nobel Prize was about, the intercommunication. And what you have here, what you see here, is via signal, how these proteins at the top, they go to specific lengths, because they are destined to a specific function. And then the 2013, which is what exosomes is about, basically, they kind of looked at how cells communicate, how there is kind of packages that are carried from one cell to another, which is what exosomes is, and basically they carry proteins. And these proteins inside the cell have the address where the exosomes are going to go. So if these proteins are unfolded or destroyed, the cell has no direction as what to go. It's like a FedEx agent with a package, but doesn't have an address, doesn't know who to deliver it. So this is what the importance of proteins. Now, if you talk about exosomes and how they carry proteins, you're talking about intercommunication. So by definition, you cannot go back to the theory of filling in the evaporating liquid. You have to start thinking about interactions. If you think about exosomes and you use exosomes, you must think about how these cells and how these proteins are interacting. And now, so increasing collagen is one small part of rejuvenation. The most important part is the like you have hormonal balance. You have protein. What hormones are proteins? But you have a proteostasis. That means all of these. There is like a balance between all of these proteins. And the proteostasis is the one that basically repairs and refolds proteins, because if the protein is unfolded, it's completely useless. So this is the proteostasis network. There's a lot of articles, a lot of books, including my book that was just finished, and it's actually in print. And this is my book here, and it's coming very fast. And also I have a lot of these concepts in my previous books that have been translated in 12 languages. Now, for skin rejuvenation, we need all of these factors. Cellular processes, response to stimulus, macromolecular metabolism, many different communication aspects, not just the collagen replacement. And why do we age? What is the main problem of why we age? Is body desynchrony. The body desynchronizes. And that's what my book is about, is the rhythm of youth. Why? That means bringing the body back in the rhythm of youth. So anti-aging is about balance and synchrony. And it's, now there is a new method that I would like to introduce with you. It's called SFRED. You can find FRED everywhere. There's a lot of articles about it. It was discovered in the 1950s. And it's resonance. What is resonance? In order to produce a resonance, the two signals must be in sync. Remember, I was talking about in sync and how there is a faster delivery of communications. In other words, you cannot multiply apples with oranges. You cannot multiply oranges with oranges, otherwise you don't get a multiplication effect. And so SFRED is body synchrony, which is health. So health is basically synchronization. And here, what you see here is under the microscope, you see how this SFRED energy, basically, it gets the cell to change its communication. And so cellular energy transfer via resonance, this is what this is. And what you see here, you see how the cells communicate. So how can you say that, oh, I can fill in the glass with some more stem cells or some more collagen or another protein and everything's going to be okay? It's not going to be okay because look, they're communicating. So there is an important aspect of that. And this is a chain reaction. Of course, this is not under the microscope. This is just a graphic, but just to show you how it basically happens. But this is what resonance is. You see how the cells react, and this is under the microscope. So the other problem is that there is no mechanism when you inject anything, really, whether the proteins are going to be folded. Because if they're not folded, they're useless. So this is basically heat will denature proteins. So if you do anything with heat, the proteins will be denatured at the same time. So you have to be careful about that. So when they're denatured, they can be renatured only with resonance. And there is a lot of different resonance techniques now. You can actually, there's a lot of this research. You can find it on Google. Or Google Scholar is full of it. So this is a functional protein right there. It's folded. When it's unfolded, is denatured, means it cannot work anymore. It's like somebody who has no brain. It's like somebody with a brain damage. They cannot work anymore. And this is different methods of folding different proteins, and protein folding can happen with signaling resonance. And actually, the greater the resonance, what you see, the proteins are getting folded faster. It's very interesting. And this is the different resonance methods, nuclear magnetic resonance, small angle X-ray scattering, and many different types of resonance. We have the signaling resonance. This is me in our kind of investigation unit in our research center, and that's just another person that works for us. And this is how we basically have analyzed the signaling resonance transfer, how it restores not only protein function, the protein folding, which will restore the protein function. And that's another picture of mine in front of the thing. This is all the references. You can see I created this lecture on the basis of a lot of references. I got them out of my book as well. So it wasn't only this lecture. I have a lot more. It's about 500 references. And thank you very much for kindly attending this lecture. This is my website. If you have any questions, science@ielios.com. I'll be very happy to answer any questions. Thank you.
Okay, so thank you,
Why
Dr.
is it
Sofra.
recording
So we'll move to the next speaker, who is Dr. Narasin from South Korea. Dr. Narasin is a leading dermatologist with over 20 years of experience specializing in well-aging treatment, rejuvenative aesthetics, and dermatologic functional medicine. As the founder of the Institute for Functional Medicine of Korea,